So good morning, everybody. We're back. We're going to start our lecture in a moment. So as we know today, we're going to continue our lectures on the, so just a moment, I think I'm not moving. Oh, I got it. Is it? Okay, it is. So we're going to continue our lectures on cell biology, and the title of today's lecture will be the cytoskeleton and cellular motors. So we're going to discuss the structures that ensure the proper functioning of the cell and maintenance of the shape of the cell, and actually even changing of the shape of the cell through different physiological processes. So let's talk about a cytoskeleton. What's a cytoskeleton? Cytoskeleton is a skeleton of the cell. So it's composed of these fibrillar or fiber-like proteins that are located inside the cell, and they form a meshwork or scaffolding inside the cell that maintain the cell shape. They're anchored to the cell membrane, as we see here, and then they will transverse the cytoplasm, ensuring that the cell maintains its own shape. So cell is not squishy, cell has inside proteins that enable and ensure the cell shape. So what are the cytoskeletal proteins we're going to talk about today? We're going to talk about microfilaments, which are here, as you see here in the bottom, the green one. And these are mainly actin filaments. They're globular proteins that are aligned in they form a double helix. So two lines of these globular proteins will wind around each other. The next one, microtubules, which are much larger. They're also formed by these globular proteins. However, they're much bigger, and they form tubes, hence the name microtubules. Next are intermediate filaments that are here marked with a yellow color, and they're formed from fibrous proteins and proteins that form strands. Next group are accessory proteins, and these proteins will actually regulate the length of these fibers. They will regulate the position of these fibers, and how do they associate with different intracellular structures, such as organelles or cargo. So we look at a cytoskeleton, and we see here a nice staining of the cytoskeleton. We can divide these cytoskeletal proteins into two groups, into shapers and movers. Shapers are formed by protein filaments, and these shapers enable the variable shape of the cell, and they also work as skeleton. You know what does a skeleton do? Is attachment point for our muscles. So the skeleton is important also in our movement. So these shapers not only regulate the shape of the cell, but they also are very important for the movement of the cell. These are made by polymers. We mentioned those globular polymers, and these polymers are made for protein subunits, those globular proteins. Now, three groups we're going to mention today again are microtubules, which are largest. They're around 25 nanometers. Next one on the size, below them are intermediate filaments, and the smallest are actin filaments. Now, movers enable the movement of the content inside the cell, and also they enable the movement of chromosomes. They also enable the movement of genetic material, and they enable the movement of organelles. And they're powered by the breakdown of adenosine triphosphate because it costs to move things to the cell. It actually you have to use gas to move from Lillington to Raleigh. So these things are actually require utilization of ATP. So these ones are known as kinesin, dynein, and myosin. So let's look at the microtubules. They're really cool. Look, they look like tubes. They're the largest of these fibrillar proteins we mentioned, skeletal proteins. They look at the cylindrical tube, but they actually are formed from these protein heterodimers. Two units. Heterodimers mean there are two units attached to each other. Dimer, two units. Hetero means two different units. So they're formed by these dimers, protein heterodimers that are composed of alpha and beta tubulin molecules. And these heterodimers is assembled in these large protofilaments, long filaments. And these now protofilaments will stack next to each other, forming a tube. So that is those are the microtubules, and they're very important for movement of chromosomes during cell division. They will form the mitotic spindle. They're involved in the process of mitosis and meiosis. They're very important in formation of cilia, those hair-like projections on top of the cells that will move, for example, mucus in your lungs. Important for formation of flagella, which is end tail of the spermatozoid. And they're involved also in intracellular transport. They're similar to actin molecules, but they're much bigger. Although you see when we discuss it that they very much are metabolized like actin molecules, smallest one. So how are they built? They're constantly growing and shrinking. This is kind of cool. They're constantly be built and destroyed in a cell. It's a very active process. And this is known as dynamic instability. So how they are built is first, the tubulin monomers, alpha tubulin and beta tubulin, will join together. And they will become tubulin heterodimer. Alpha tubulin and beta tubulin. Hetero means two different proteins, dimer, two proteins. Now what they're going to do is that when they bind, they will actually then bind to GTP, guanosine triphosphate. And now these heterodimers will bind to each other and form long protofilaments, we see here. But there is a catch. As they are inside the protofilaments, what's going to happen is that the GTP will be broken down into the GTP, guanosine triphosphate, into guanosine diphosphate, and they will be kept in a center. However, the top, the positive end of the protofilament, will actually still have a GTP attached to heterodimers. And this is known as a GTP cap. It's very important. And now what's important for these microtubules, they will continue to grow and grow until they reach an anchoring point, until they reach, for example, an organelle or everything else, and then they will stop growing and stabilize. Now, important for them is that if they do not reach the stabilizing point, they don't reach something they need to hold on, they will actually fall apart. So this is the characteristic of, and how do they break down? Addition of GTP and alpha and beta tubulin dimers, heterodimers, will slow down. What's going to happen? We know inside the protofilaments, the GTP is broken down to GDP and phosphate. Now, this breaking down of GTP to GDP and phosphate will catch up and will completely destroy a GTP cap. So top now will be attached to GDP, not GTP. So that cap is gone. So now the new heterodimers will not bind because they usually bind to GTP-bound heterodimers. So they will not bind to them. So structure will become destabilized. These protofilaments will peel off like a banana peel, and they will fall apart. So while the new tubules are constantly being assembled, and they constantly radiate from the structure we're going to mention in a second, the centrosome, the old ones are constantly disassembled. Now, the moment when this growing microtubule reaches an anchoring point, it's going to be stabilized, so it will not break down, by the action of specific group of proteins which are known as stabilizing proteins that prevent microtubule disassembly. Now, this is a centrosome, and we haven't mentioned when we describe the organelles because some people put centrosome in organelles, some people put in just intracellular structures. But what is the centrosome? Centrosome is the star-like structure that's actually found and seen in cells that are not under mitosis, they're not dividing. They're found on one side very close to the nucleus. Stay like a star on top of the nucleus. So what's their function? They are anchoring point for microtubules. They're involved in organizational microtubules based on cellular requirements. So they regulate number of tubules, they regulate orientation and cytoplasmic location of these tubules. So microtubules will irradiate from the centrosome like star pattern out of it through the cell. And of course, microtubules will be constantly growing and shrinking. So they're under constant dynamic instability. Now, this is the important role of microtubules. The important role is formation of mitotic spindle. Microtubules will have to separate and split genetic material during division. So they will push and pull chromosomes in dividing cells. They will, by doing this, segregate the genetic material, and they will start working from centrosomes. So let's look at this. We'll see that at the beginning of mitosis, when the cells has just one nucleus, nucleus will fall apart, nuclear wall will fall apart, and all the microtubules will be disassembled in a cell that's getting ready to divide. Then microtubules will reassemble and will form mitotic spindle between now two important structures and two centrosomes. Now you're going to have two centrosomes in the end, and they will now connect with each other with microtubules. This is the formation of mitotic spindle. Now, microtubules, as they grow from one centrosome and go to the other, they will attach to chromosomes and bind. So these microtubules are known as polar microtubules, and the chromosomes will have these things that look like cat's whiskers, okay? So these are known as kinetochore microtubules that will bind to the polar microtubules spanning from one centrosome to another. And the last group of microtubules are astral or aster microtubules, two names. I call it, there are books that call it astral, some aster, they all go from the star. So what they do is they just spread from the centrosome throughout the cell. So what happens is, microtubules, as they grow and form the mitotic spindle, they will bind to kinetochore microtubules on the chromosomes. And then as they grow, they will align chromosomes and they just tear them apart. So that is the process of microtubules and their importance. Why is this important clinically? Because of this. We're going to talk about mitotic spindle poisons. These are the drugs / poisons. What they do is they prevent the formation or breakdown of mitotic spindle. So when mitotic spindle doesn't break down, cell doesn't divide because the spindle remains, cell doesn't divide. Or if it if the mitotic spindle is not formed, cell will not divide whatsoever. So this first one I'm going to mention is colchicine. It's actually a drug that is used for treatment of gout. And what does it do? It halts and inhibits the cell division. It stops the cell division, especially immune cells in this situation, because it binds to unpolymerized tubulin, so it prevents the formation of microtubules. What does it do? It binds to unpolymerized tubulin and prevents the tubulin assembly. So if you have rapidly dividing cells, they will start breaking down, and their microtubules, mitotic spindle, will actually break down in rapidly dividing cells. Very similar to this is activity of certain anti-cancer drugs. You don't need to know the symptoms or what do they treat, but I wanted to show you the relationship. So now, the drugs that use this in vincristine and vinblastine. Vincristine is very much used for treatment of non-Hodgkin lymphoma, and very important is leukemia, acute lymphoblastic leukemia. And also, it's for treatment of cancers of kidneys. Vinblastine is very much used for testicular cancer or bladder cancer. But what's going to happen? Once you affect the mitotic spindle, what's going to happen with mitotic spindle in normal cells, specifically rapidly dividing cells? They will be affected too. So they'll have a lot of side effects. So you know that's normal. They will cause neuropathy, inflammation of nerves. They will cause loss of sodium from the body, constipation, headaches, a lot of other things. Another mitotic spindle poison is Taxol. Taxol is actually the substance that is used more experimentally, but it's also used as an anti-cancer drug. So what does the Taxol do? It actually binds to polymerized tubulin, so it prevents the breakdown of tubulin. So think about it. You're a cell and you're dividing, and you can't break down your microtubules. That means that you can divide, and cells will be affected. So they prevent microtubules disassembly and arrest the dividing cells in mitosis. They really cannot break apart from each other. So one of the good examples is paclitaxel, which is very effective use in treatment of breast cancer. Also melanomas. And on the boards, don't need to remember this, I'm just putting it so you see the connection, they're used extensively for treatment of Kaposi sarcomas, which is a very rare form of cancer that is seen in HIV patients. Of course, these drugs also affect your normal cells. Patients will lose hair because hair divides rapidly. Patients will have joint pain, problems with their production of the synovial tissue, and they will also have nail discoloration, which will last forever. So they will have discolored nails. Now, microtubules also form cilia, and flagella. So what are these? These are really cool stuff. As you know, they're formed by stabilized microtubules. They don't fall apart. They're stabilized. So cilia are found in your respiratory system. You know that cells that line your respiratory system have those little hair-like projections. Those are cilia made from microtubules. And they will move in one direction, towards your mouth. They want to move a mucus and content out. So they will move in cyclic motion, in cyclic strokes towards the mouth. And they ensure that there is an adequate movement throughout the respiratory tract. Flagella is actually found, and they're much longer, and then cilia, and they're found as a part, and they're part of the spermatozoid tail. So how does the flagella, how does the spermatozoid tail look like on a cross-section? So you see here 9 pairs of microtubules that form a ring around the central structure that has 2 microtubules inside. That's actually the spermatozoid tail. And now, how is the movement achieved? Is by these microtubules bending and sliding against each other and moving the tail. The tail usually goes in rotating fashion. Okay? And there is a protein that makes bending and sliding and ensures bending and sliding of microtubules, and this protein is known as denane. So this protein, as we see here, looks like a horseshoe on the sides, is actually the one that ensures the bending and moving of the cilia. Now, we go back clinical. Why the cilia is important? We know cilia are important. No, sorry, in this situation we talked about flagella, but it's the same thing. So what's a characteristic for the cilia and flagella, and what is the importance of their clinical importance? As the embryo develops in the mother's womb, we know that organs will be developed in one location and they will be shifting in another location during embryological development. So all the organs, most of the organs will be actually shifted during the embryological development to their position. This is achieved by the presence of cilia inside the embryological tissue. So if the individuals have problem with the movement of cilia, they can develop a condition known as situs inversus. Is where organs are flipped. They are not in the positions where they are. Situs inversus totalis, a complete inversion of all the organs, is rather rare. What you're going to see is usually dextrocardia, where the heart is on the right side, not on the left side. So that is the characteristic, and if you are unlucky during your rotations and you have a really nasty attending, they can actually give you an EKG of this person, and you'll have a nightmare, okay? Because now it's measuring something totally different. They're all off, okay, leads. But that is a characteristic. Now, this condition occurs in very rare, in 1 in 10,000 people, but they're associated with something called a Carter-Jenner syndrome. So what's a Carter-Jenner syndrome? We're going to mention in a moment. But we know that about 20% of individuals that have situs inversus have a condition known as primary ciliary dyskinesia. What does that mean? Abnormal movement of cilia. So they have a dysfunction of the cilia, and of course this will manifest during the embryological phase of development, because we know that cilia will move these content into the directions, okay, in appropriate location. So also important to mention is that in patients with primary ciliary dyskinesia, around 50% of them have chance to develop situs inversus. Not everybody develops it, but around 50% of them will have situs inversus. These individuals will have something called a Carter-Jenner syndrome. Carter-Jenner syndrome has a triad. It has situs inversus. All the organs are in opposite direction, or some of them. They're going to have chronic sinusitis, and they're going to have bronchiectasis, which is the accumulation of mucus inside their lungs that does not move. Why is that? Their cilia are not working. So the mucus that should be eliminated stays in that area, and it's very susceptible to infection. So these individuals develop these large bronchi full of mucus. Okay? They will have problems with sinuses because they will not be able to drain sinuses. More important is that males who suffer from Carter-Jenner syndrome, very common question on your board, will actually also be infertile. Why? Because they will have problem now with the flagellum, with the tail of the spermatozoid that works very similar to cilia, and they will have their spermatozoids will not move. However, microtubules act as an anchorage point for something called the microtubular motor proteins. What are these? We understand that some of the movement through the cells of the content and mitochondria is very much dependent on microtubules. They're staying there. They're the skeleton. However, they can actually help movement of content along the cell, inside the cell. How? Because there are microtubule binding proteins, specifically motor proteins. We mentioned dyneins before. So dynein and kinesin, which will attach to the microtubule on one side and attach on, for example, in this situation, mitochondria on the other side or any content inside. And they will walk along the microtubule, pulling that content. Okay? So these are the dyneins and kinesins. What do? They actually have ATP binding heads and tails. And then they have the area, the other part of their molecule that attaches to the cargo. So they will break down ATP into ADP, which will enable the movement and the further attachment to the microtubules. So pretty much walk and pull the content through the cell. So dyneins will move towards the centrosome, so towards the center, while kinesin will move away from the centrosome. And of course, what provides energy for their movement? ATP, breakdown of ATP. The next group of molecules I want to mention are intermediate filaments. They're smaller than microtubules, but they look like ropes. They're actually very strong. And these intermediate are larger than actin myofilaments, smaller ones, but they're actually larger than microtubules. They're actually smaller than microtubules. So they're in between the microtubules and actin size. Where are they found? They're found in cytosol, and they found actually between the nuclear envelope and plasma envelope. They form a network from the nuclear envelope all the way to the cell membrane. And their function, of course, is to provide the stability inside the cell, specifically maintain the cell shape. And also, there's a group of the intermediate filaments that are known as nuclear lamins that provide the strength and support to the nucleus. They're inside the nucleus, and they provide strength and support. So what's their structure? Look, they actually are formed by the alpha helical rod-like protein, you see here, and two of these proteins will wind around each other. And they will result in a formation of dimers. Two proteins wound around each other. And these are known as coiled coils. Okay? Now, these coiled coils will self-associate in a staggering pattern to form tetramers. They will form these 4 tetramers inside. So they will bind, these coiled coils will bind to each other, okay? And they will form ropes. And they will form 4 small ropes, okay? So these are tetramers. So, and coiled coils are arranged inside tetramers in anti-parallel orientation. They are not organized in, they're one that is on top of the other, looks like a mirror image. Now, these tetramers will attach to another tetramer, side to side, and they will actually form now 8 bigger ropes, okay? And these are known as, so they will be 8 tetramers that will combine and wrap around each other, forming a very strong rope. And eventually, you are getting the intermediate filament. So very, very strong structure that has, first it starts with dimers, coiled coils. Coiled coils attached to each other, forming now tetramer inside 4 different strands. Now these 4 different strands will now bind all together to each other to form 8 strands, and these 8 strands will wind around each other. You can imagine how strong that is. That's a really strong measure. So this is just a list of different intermediate filaments and what do they do. So it just tells you that they're involved, for example, providing strength to the cell. They're very much involved in providing strength or support to neurons, such as nestin, which is very much involved not only providing strength to the neurons, but also involved in the regulation of growth. We mentioned nuclear lamina by the type 5 of intermediate filaments who are involved in maintenance of the structure of the nucleus of the cell. Actin. Actin is the smallest of these guys, and actin is present in cytoplasm and nucleus. Actin is all over because it is small, It actually provides a structural support to these. It radiates from the cell membrane, and what does it do? It forms the microfilaments throughout the cell, these tiny, small filaments that are inside the cell and provide support to the cell. So what is the result? They will actually form a network that will span all the way from nucleus to the membrane, and importantly, there is tons of actin just under the membrane. So this actin supports membrane, okay? So it supports the structural support, provides structural support to the membrane, and this area is known as cell cortex. So it is the most dense area of actin just under the cellular membrane. Now, what does actin do? It regulates cell contraction. It's involved in the process of contraction. But it also regulates the expression of genes in nucleus. So it's involved in chromatin organization. So let's look how it looks like. So it's actually formed by G-actin monomer units. Single globules, G-actin monomer units. And they will form two strands that will wind around each other, forming an F-actin strand. Okay? So that is actually the actin strand. So G-actin monomers, these tiny little globular proteins, will bind to each other, forming two strands, and these two strands will wind around each other, forming the F-actin. Now, how this is done is free G-actin monomers are first bound to ATP during lag phase of formation. Then 3 G-actin monomers that have all of them attached ATP will bind to each other, 3 of them, and form a preliminary F-actin molecule. From them, the F-actin molecule is going to be started to produce. This process is known as enucleation. Then new G-actin molecules will be added to the positive end of this F-actin molecule, growing F-actin molecule. And this is known as polymerization. There is an add of new G-actin molecules on the positive end of the growing chain. Now, inside the, and you remember this, inside the, very similar to microtubules, inside those F-actins, the ATP attached to G-actin will be hydrolyzed to ADP, okay? And this is going to happen during this polymerization process. At the negative end, the G units that are attached to ADP will be removed from F-actin, and at the same rate as the new ones are added. So what does that mean? Is that if I'm F-actin, I will grow and grow and grow and reach a certain length. And then what I'm going to do is I'm going to continue growing and continue losing G-actin, okay? So it's a constant active process. And now when you look at it, this process of adding new G-actin and removal of old ones are known as treadmilling, like a treadmill. What does that mean? A G-actin gets attached to the growing F-actin, then it's being pushed further down by new G-actin coming in, and then it will lose its ATP into the ADP, and will eventually reach the end of the line of the F-actin, and it will be removed. So it almost looks like the G-actin is moving down that F-actin. This is known as treadmilling. Bless you. So now I'm going to talk about the poisoning. I remember in my time practice, I actually seen that. I was still actually an intern during this period, but one of the characteristics is, one of the cases you're going to see on the boards, and maybe in your practice, not as much, is you have a group of kids who went into the forest to play, and they come back and develop serious problems. Why? Because they were cooking lunch using local mushrooms, okay? That's what I had, okay? So I don't know whose kid was that, who had that idea, but I want to talk to their parents. However, these are not as common, but they're a good example of problems with actin. So first, we're going to talk about Amanita phalloides, and that's important. These mushrooms have an awesome name, very attractive name. It's called the death caps or angel of death, okay? So what happens is, these mushrooms contain, and they actually in Europe look like edible mushrooms. That's a problem. But they have 2 toxins in themselves. They have amatoxin, which inhibits the RNA polymerase. And you're going to hear about that further down. But more important toxin that I'm going to talk about is known as phalloidin. Amanita phalloides is the name of the death cap. So phalloidin is the toxin from that mushroom. And what does the phalloidin do? It disrupts the normal actin formation. What does it do? It binds to F-actin much stronger than G-actin. The toxin binds to the F-actin. It makes it much stronger. It prevents the breakdown of this F-actin, and it also prevents the breakdown of ATP hydrolysis inside. So you remember actually the G-actin continues to grow and grow. No, F-actin continues to grow and grow and grow and grow, and it doesn't get broken down. So eventually you have a cemented cell full of these F-actins, and these cells will die. So this abnormal growth of the F-actin will inhibit the cell movement, and will inhibit both intracellular and extracellular cell movement. So what you're going to see? You're going to see a patient who ate these mushrooms, and for a couple of days they will be okay. Then they're going to show up in a practice with serious GI problems, diarrhea, vomiting, and everything else. Then again they'll be okay for 4 to 6 days. They'll be fine. Takes time for all these F-actin to fill the cells. Okay. Then after that period, they will develop liver and kidney failure and end up in intensive care. Okay. And the therapy in late stage of poisoning is usually liver transplant. Okay. So what happens is that the liver cells will be affected. Why? Because liver cells are the ones that are processing the toxin. Okay. So they will be one affected. The therapy is detoxification. Again, if it doesn't work, usually these patients end up with having to have liver failure and end up doing liver transplant. Let's talk about actin-binding proteins. Actin-binding proteins are proteins that modify actin. So what they do, they can cross-link actin. They can make actin much tightly together. Okay? Or these proteins can break down actin. They can chop this poor actin and make it fall apart. Okay? So those are the actin-binding proteins. So those are the proteins that can modify F-actin in one way or the other. So they can prevent polymerization of F-actin. They can stimulate polymerization of F-actin. They can cross-link F-actin. Why they are important? They are one of the importance of these actin-binding proteins are that they are involved in gel-sol regulation. What's a gel-sol? You know that cell inside and cytoplasm can have two states. It can have gel state and a sol state. We know what the gel state is. It's a jello state. It's much thicker, much denser cytoplasm, or cytoplasm can go in a sol state, which is solubilized state, when it is more like water. So these actin-binding proteins will control that, because we know actin is inside the cell all over. So the gel state is more dense, and it has more dense and more structured actin inside it, much more tightly bound together. So these proteins will cross-link F-actin molecules, make them connect to each other, and make them packed much more densely. Now, the sol actin-binding proteins will cause the cytoplasm becomes less dense. So what does that mean is they will fracture and fragment actin. They will break down that actin and make the consistency of the cytosol or cytoplasm much more watery. So they will fragment F-actin filaments and also inhibit polymerization. They will prevent the formation of new ones. Two ones I want to mention because they pop out on your boards. Coffilin forms twisting and fragmentation of actin. It twists and then fragments the actin, while gelsolin will cut away those cross links between different actin molecules. So actin molecules become fragmented, they become loose. One of the things I want to show you is also that these proteins, these actin-binding proteins, specifically the ones that make cytoplasm go into the sol phase, require calcium for their work. So calcium is quite important for their activity. So let's talk about the actin-binding protein. Clinically, I'm going to mention spectrin. What's a spectrin? Spectrin are these long, flexible dimer rods, and they impart the strength of F-actin. So what do they do? Best way to explain is this. they bind and strengthen F-actin filament. You know where F-actin is? You remember cell cortex? There's a lot of actin under that cellular membrane. So these spectrin molecules will cross-bind and strengthen this area just under the cellular membrane. Makes sense? So they maintain the shape of the cellular membrane. They cross-link those F-actin molecules just under the membrane. They also bind two different proteins, which you don't really need to know, but you need to know their name because they'll ask you on boards. They'll bind ankyrin and protein 4.1 together that are actually parts of the membrane. They kind of bind them, and the one is on one side of the membrane, another one on the other, and as they bind them, they maintain the strength of they work almost as bridges to maintain the strength of the cell. So they facilitate their interaction, and we see here is our good old spectrin. Look, it's lining the inside of the membrane, and now what's attaching to it are actin molecules. So it almost acts as anchorage for actin molecules that will support that and strengthen that membrane. Why are they important? Specifically for maintenance of big concave shape of your red blood cells. You remember that your red blood cells have two concave shapes, and that ensures that they have much larger surface area to transport oxygen, number one. Number two, what happens with your red blood cells when they travel to your body? They go through very narrow capillaries. So what they have to do? They have to squeeze through those capillaries. So the membrane has to be very elastic. Makes sense? And that is what spectrin ensures. Ensures their shape and that they're elastic. And also, it is important because it maintains its shape and increases the oxygen capacity of the red blood cells. So condition, disorder is hereditary spherocytosis. It's a condition where there is a spectrin deficiency of abnormality in these individuals. They might have abnormal spectrin, or they might not produce enough spectrin. It's autosomal dominant hereditary disorder, so you see that it can be carried easily. It's a very common question on boards, and you might see this. This is not that rare. And one of the reasons on the boards, they will ask you what is the reason for hereditary spherocytosis is a lack of ankyrin protein 4.1 interaction. But not only that, it is that there is no spectrin in it, and the spectrin anchors actin, F-actin molecules. So the strength of the membrane is appropriate. So what does it mean, hereditary spherocytosis? Spherocytosis, hereditary means it's inherited. Spherocytosis means that the red blood cells are spheres. They are not concave. They make nice circles, okay? And that is not good, okay? Number one is that these red blood cells can carry less oxygen because they are not big concave. The surface area is much smaller. So when you look at it, if you see on these images, you see these red lined one, red nice round ones, these are spherocytes. Because normally red blood cells have paler in the center. Why? Because they're biconcave. So you will see that concave has a paler in the center. They look like little rings. That's a normal one. So what happens with these cells is, number one, they don't carry enough oxygen. Number two, they're round, so they're a little bit bigger. And number three, when they get stuck, they burst. Okay? So these individuals will actually develop hemolytic anemias. They will be losing, their red blood cells will be breaking down like crazy. Specifically, when red blood cells go to an oil filter of the body, and not oil filter quote unquote, to filter of the body known as spleen. So these big, large red blood cells will get stuck in the spleen and they will break. And these individuals will develop anemia. So what are the patient symptoms with this disorder? Is they will develop hypoxia. Why? They feel they don't have enough oxygen. Because they don't have enough oxygen, because these spherocytes can be destroyed and they do not carry enough oxygen with them. Okay. They will develop hyperbilirubinemia. They will develop jaundice. Now, for all of you guys that didn't have biochemistry, the breakdown product of hemoglobin that is inside red blood cells is bilirubin. So if they're breaking and bursting, they release a lot of hemoglobin in the blood, and this hemoglobin is now converted into bilirubin. It's a breakdown product. And this bilirubin will accumulate in their tissues. So they will develop jaundice. Okay? And they will develop gallstones. Why is that? I love physio, so we're going into physio. Is this? They have high amount of bilirubin. This bilirubin is excreted to through the bile. Okay? So they get tons of bilirubin inside their gallbladder, and this bilirubin will form gallstones. So the therapy, one of the interesting things is you're going to see a patient who develops sudden, doing okay with the hereditary spherocytosis, but suddenly develops infection. Now the number of these red blood cells bursting is going to increase, and this is known as hemolytic crisis. The therapy is to remove a filter. Okay? You can't really solve the genetic issue, but what you can do is get rid of their spleen. So the filter is gone, and then there's going to be less breakdown of these abnormal cells, and of course remove their gallbladder because you don't want them to develop gallstone. Next one of actin-binding proteins is dystrophin. It's a very big thing to me, simply because I spend a lot of time during my practice volunteering for a Muscular Dystrophy Society. And dystrophin is associated, impairment of production of dystrophin, which is actin-binding protein, is associated with muscular dystrophy. Let me explain it. So what does the dystrophin do in a body? What does it do? It cross-links and straightens actin molecules, makes the actin much stronger. Okay. There are two ways that this dystrophin works. Number one, it attaches intracellular skeleton of your epithelial cells. You know what epithelial cells are? Cells that are on the surface, for example, of your bronchi. Okay? So they're there, and they attach to area below them called as basal membrane. You have just one layer of epithelial cells, for example, and then you have a basal membrane in the bottom. So they attach to that basal membrane. That's where they hold their behinds to, okay? And then they perform the function. So what does the dystrophin do? Ensures the attachment of these cells to the basal membrane, okay? Because it cross-links and attaches the actin inside with the basal membrane through the cell membrane, okay? That is the characteristic of it. Second thing, what does dystrophin do? Is actually forms the dystrophin-associated protein complex. Look, this is the cell. Specifically, this is a muscle cell. It's very common in muscle. And what does it do? This is actin, filaments, and they attach to dystrophin. And then dystrophin attaches to the membrane. So what does that tell you? Is they attach F-actin to membrane, strengthening the membrane in muscle cells. Okay? Also, the result is formation also, and they also work partially to send signals from outside into the cell, but more important, they provide tensile strength for muscle. So individuals who are born with muscular dystrophy are actually individuals who have lack of that protein known as dystrophin. There are two ways. They might have abnormal dystrophin. They might not produce dystrophin whatsoever. Okay, so there are two different reasons. So usually, they will have problem with production of dystrophin. This cross-links the F-actin, specifically attaches them to the membrane of the muscle cells. So what's going to happen with their muscle cells? Muscle cells are constantly injured, are they? They're constantly under stress. So muscle cells will get destroyed through any kind of activity. As they get destroyed, the muscle cells are now replaced with connective cells. So suddenly, muscle, nice red muscle, becomes a connective tissue, tendon. Okay? So that is a characteristic of the muscular dystrophy. So eventually, the muscle tissue is replaced with connective tissue. Why it is? Because as you move the muscle, you will, of course, you will actually provide tensile strength to that muscle. Muscle contract and relax. And now, they don't have dystrophin that will not support these cells, so the cell membranes or cells will get damaged. They get damaged, they're replaced with connective tissue. One of the characteristics of muscle dystrophy is something I want to mention to you, is pseudo-hypertrophy of calf muscles. I've seen that in kids. So what happens is that before your kids will even show signs, large signs of muscular dystrophy, their calf muscles will be enlarged. Okay? Their soleus muscles will become much bigger. They look like they're really exercising, okay, their calf muscles, but they're not. The calf muscles are the first one to get broken down, and calf muscles are replaced with fatty tissue. So pseudo-hypertrophy means looking like enlarged muscle. It's not an enlarged muscle. Two types, Duchenne muscular dystrophy and Becker's muscular dystrophy. Let me explain you. Duchenne's muscular dystrophy is more severe. Why is it? Is you have genes. You just talk about transcription and translation today. And you have codons. okay? And did you mention point deletions? So muscular dystrophy, Duchenne muscular dystrophy caused because of point deletion, 1 or 2 of the purine or pyrimidines are lost. Not the entire gene, just portion of that gene, portion of that codon, portion of that triplet, okay? So what happens is that they will develop now frame shift mutation. So the whole reading will be shifted for 2 or 3 purines or pyrimidines. So what's going to happen is that they will actually develop a very weird protein. They will develop the protein that is usually very short because they will reach a stop codon and eventually stop. And what's going to happen is they will produce non-functional dystrophy, so they have more severe disease. The second one is Becker's muscular dystrophy, which is less severe. And you know, my patient used to say, oh, he's lucky, he's got Becker's. But what the Becker's is that they're less severe. Why? Because they will produce a dystrophin protein. It's just not gonna look as normal. Okay, it will not perform normally. So they will produce short but functional dystrophin. And they will have, why is this happens? Because they're larger deletion. What if you lose 1 or 2 triplets? 1 or 2 amino acids of a protein? That's fine. You're going to be still reading it. You're just going to be missing 1 or 2 amino acids. So this protein might still work. But that's the characteristic of this. Few things as you remember is that it's a recessive disorder, which means is that actually that one of the kids will develop the disease, males. It's X-linked recessive. Now, let's look at the Gover sign. Gover sign is one of the things that I just want to mention is that when you see and identify these kids, you look at them, they will actually use their hands and climb as they try to stand up from a sitting position from a floor. They will use their hands to climb on their thighs in order to straighten up. That's called the Gover sign, just for your information. Now, I have a brief case. Please don't send me emails, Dr. D, what does this case have to do with the lecture? I got that last year. Okay, let me explain you, but you will know. We have a case of 45-year-old male who works as a high school chemistry teacher, okay? And a part-time crystal meth producer who present with general weakness, fatigue, and dizziness, and symptoms have started a few months ago, but are becoming worse. He's becoming weaker, dizzier, worse. Patient also lost significant amount of weight due to the appetite loss. He also stated he has been suffering from a number of infections in the last few months. Good question. If your patient tells you they're a crystal meth dealer, what do you do? No, you don't. If he tells you he's going to kill himself or everybody around him, then you call cops. But if he tells you he committed a crime, you as a physician are not reporting that. Okay? That goes into your patient-physician interaction. You don't, okay? But this is it. So that's why you know he's dealing drugs, and you watch the series, so you know exactly what he's doing. But this is it. So clinical exam will relieve presence of bleeding around the skin, petechia, bruising, and large lymph nodes and peripheral edema. There are large lymph nodes. He's swollen. Radiographic studies are unremarkable, and then you put this patient and look them under laboratory analysis. You will ask this patient to do bone marrow biopsy, poke the bone marrow and look inside to see what's happening with his red blood, with his blood cells. So blood smears will show a large number of leukocytes and the presence of these immature, hard, big, blobby lymphoblastic cells. These cells are normally not found in blood, okay, because these are immature white blood cells, okay? But you're suddenly seeing them all over his body and all over there. So, of course, you refer patient to oncologist, and he's subjected to treatment protocol that consists of vincristine, daunorubicin, asparaginase, and dexamethasone. So why is this case important for you? Vincristine, that's a drug, if you remember, it's a mitotic spindle poison. Did I get it? Okay. So please don't send me emails. Everybody's like, oh, you're talking about leukemia. Are we going to need to know leukemia? No, you don't. It's just an example. So, you know, in the future, you'll know why you're giving this patient the vincristine. What you want to do is you want to prevent the rapid division of the malignant cells by affecting the formation and the breakdown of its mitotic spindle. So the patient is diagnosed with acute lymphoblastic leukemia. So the large lymphocyte count actually indicates that he's not going to do well. So what's the most common cause with people who cook meth is benzene. They're exposed to benzene during this, specifically if they use it as a solvent for the production of crystal meth. That's why they try to wear those masks. And the bone marrow biopsy will provide a most accurate diagnosis. We're going to poke their bone marrow and look inside. And just before I let you go, just one experience, what really attracted me to the muscular dystrophy, what kind of stuck in my head when I practiced and I actually volunteered for Muscular Dystrophy Association. I was volunteering for an institution where people with muscular dystrophy live and work. And that's how they pay their upkeep. And it was very important for them so they didn't depend on the government. It was actually a hospital. It's actually an institution built by people with muscular dystrophy and actually designed by people with muscular dystrophy. So just one of the things is just to tell you a story, so it's going to stick in a head, is, you know, I have a lot of friends there because I'm volunteering. I'm not their official physician. So one of them calls me and says, we have a problem. 2 of the people with muscular dystrophy have a child. Child had some genetic issues previously, so they're worried that the child is walking strange. It's walking in a strange way. So they're kind of worried whether the child is in that age, is starting to develop signs of muscular dystrophy. He was not tested. His parents never wanted him to be tested. So here I go there, I look at the kid and I says, come here, and he walks to me like this, and I'm like, okay, there's a problem. He's not flexing legs. So a friend of mine said, no, just wait, observe him for a couple of hours. So I did. I sat down, had coffee with them, watching the kid play with other kids. No problem. He's running after the ball. He's like, you know, playing soccer. No problem. I call him back. He comes back again like this. So I was like, what is this? It took me time to figure out what he's doing. He's actually imitating his dad, okay? Because his dad walks like that, and that actually gives him kind of feeling of authority. So it was right. They did a test. Kid didn't have muscular dystrophy, and, you know, he's better now. He's fine after some corrections of genetic disorders. So it tells you that you actually have to pay attention. At least it taught me. I was never thinking of it. First reaction was, okay, let's try the go-over sign. It's done. But actually, it was just the kid trying to look very important like his dad, you know, so that's it. Okay, thank you, guys. I'll see you again. Thanks.